Okay, guys, how's everybody today? We kind of think we got it set up, sort of what we want to do here. See if we can get this to come up. All right, so we're gonna run through 28 today. Other thing too, just for y'all, FYI, I'm still having some of y'all that are absolutely about to drive me crazy when y'all cannot figure out how to do a PDF. Okay, I'm gonna stand up screen in the next meeting. Because I've been thinking, like, how many of y'all have an introduction to computer? We require that, is to get our 4-year degree, get our 2-year degree, but the senior colleges don't require it. So I've had a little problem with that. But I'm about ready to stand up and say everybody needs to take the second part because y'all can't figure out how to do PDFs. It's driving me crazy, okay? All right, so if you have an Apple phone, which 75% of people have, 70% of people have Apple phone, Notes will do that. If you go to Notes, it's got a camera on Notes. Click the camera, and you can take 30 pictures and put all of them in one PDF. Then email that to yourself. Then upload it, okay? But if you need help, come see me, okay? I don't know who I'm talking to. Maybe I hit on over here, okay? Get one smile, okay? But I know y'all can do this, okay? Y'all are very smart young people. Y'all had a phone since the 5th grade, or since you was 5. Y'all can figure that out, okay? But JPEGs are big. That's why, and I cut it off. It won't take JPEGs. And I'm not going to take them by email because I got 200 students. And I'm not going to have 500 emails, but all that. Okay, so I want it in the modules and those uplinks. Okay, and if you need help, come see me. Now I will take that stuff late. Okay, pretty cool about that being late, but it'll be late to a point, right? So we're gonna have a deadline for probably next Wednesday or so for all these to the midterm because that's your notes. I'm not gonna let you do them after you fail the test, right? Oh, you can turn them in after you fail the test because they're gonna be the written part, right? Everybody with me? So I'll put a hard deadline. I'll send an email out to everybody about a hard deadline, but I would say sometimes it's probably next week, maybe next Sunday, not this coming Sunday, but the next Sunday, got to be done. That way you'll have them to study by. Okay, but it's a fun chapter today. We're finally getting into something I find a little more interesting. Sorry, but I'm just not a big fan of some of the fungi, right? Stuff I don't know much about, but animals are a little bit more interesting to me since we're in this group. All right, and let me grab a list of the questions because I'll run right through one. Here we go. So right off the very bat, we had a couple of them here. What is the evolutionary advantages that might have? And this is one you guys, these are some thought questions. So as you go through this chapter, you may have to wait and answer one at the end. I get tickled. I have somebody once like email me like, this ain't in the book. This is higher level thinking, okay? Where you have to read a chapter and then come up with a conclusive answer, okay? That's like Bloomberg Taxonomy 3. Okay, one is piece of paper, test. What's this? Piece of paper. That's memorization. Okay. And then apply, but that's an application one. So look at this one. What evolutionary advantages might there be to invertebrates having multiple developmental stages, each with a different body form, a different habitat, and different lifestyle? So that's one that's not in the book. You're gonna have to read through this and kind of get a feel for that. Okay. All right, number 2, list 3 common characteristics of animals that are not found in other multicellular organisms. I'm looking to find that one here in just a minute. Okay. And let's see what we got here. So all animals are multicellular. They're heterotrophic. So we've seen heterotrophic before. Heterotrophic means you what? Eat somebody else, right? Right? Okay. And that's what it means that they must acquire nutrients from an external source. They can't make their own food. Fungi digest their food externally and absorb nutrients. Animals ingest, eat whole food, and digest internally. So that may be one of those foods if you count for that. Animals have a variety of life cycles, right? We'll still talk about that. Many reproduce sexually, some reproduce asexually. Many animals have a diploid life cycle that's dominant, right? Remember, that's usually more common, right? We're diploid all ourselves. But here we're going to start out. We're going to come back to that question. It's actually answered in a second. Here's a cool, a bee with all that pollen on it. But animals are descendants from a single common ancestor, right? And within the animals, there are 2 big groups: the invertebrates and vertebrates. Let's define that real quick. So vertebrates are those who have a backbone. Our cervical column, right? And it can be bone or cartilage. So like sharks and stuff, do y'all know they don't have hardly any bones in them? They have the jaw, and that's about it. So they have a cartilage skeleton, okay? But it's still pretty tough, right? Invertebrates are those that don't have the bones or cartilage backbone, okay? And we'll start with those and work our way up to the others, okay? Okay, here are some common features. And there's more than 3 here, but look at them. Animals multicellular, and all animal cells like a cell wall. They're heterotrophic. Let's look at, I think the one I'm looking at maybe CB better, because there are others that are heterotrophs, right? Like fungi, right? But here's one that's unique to animals. Animals typically can move, have some type of motility. They have muscle and they can move. In other words, plants can't, like, I want to move in the front yard, right? They can't do that, right? If you come home one day and the trees have been moved around, you need to go up to Lakeside for a couple of weeks, right? Get a cycle valve, right? Trees don't get up and walk around. Animals move, and we're going to see the development of the nervous system. The grass does not feel it when you mow him or her or it. So the big two ones I think stick out real quick to me are what? Movement at some stage. Now, some animals don't move, can't move as adults. Some can't move as a young individual. So, but at some point in life, they have some ability to move, but to develop the nervous system. So as we go through these, I want y'all to look at that. Look how that nervous system develops, okay? Very interesting, all the way to the brain, right, of our animals. Most animals do sexual reproduction. You begin life with diploid zygote and then undergo development later on, and you have specialized tissues. We have all three types of the germ layers: ectoderm, endoderm, and mesoderm. So I think you can get the answer to that. I would, but definitely make sure you got nervous system and what? Movement, right? Those are the big two I think that really stick out. And we have to eat somebody. And we do internal digestion too. That's big. Okay, now you remember this group of colonial flagellants we think we come from, these cornoflagellants, or whatever, I don't know how that's pronounced, okay? But this is the protista that we think we come from. And I want you to look at this guy or critter and see if he don't look like a sponge cell, okay? So I'll show you something really wild. But here's what we think, this is where we think the animals come from. And the DNA kind of tells us this. Here is this single flagellum, okay? And they form a spear, and we think this is where the cell came from. The first multicellular, the first animals, okay? Now that's an animalistic protista, right? But if you put them, we do know they can form a colony like this. Well, when you get a colony like this is what you look like at the 32 cell stage of development. So all the animals look like, you know, mom and dad, sperm, egg come together, you form one cell, 2, 4, 8, 16, 32, around 64, 128, you form a spear where all the cells go on the outside, just like that, okay? And you have a hollow spear. Now, the next thing that's really cool is we then have one end pinches in, and this pinching in your development will either be your mouth or anus. And we're going to find out later on. That's pretty cool, and it's kind of crazy. But what we're going to find out that we're in this group called urostomes that develop butt fur. So in your mother's womb, you develop anus and the mouth later. And the only other animals that do this, it's kind of cool stuff. The only other urostomes are the echinoderms, which are the sea stars, the sea urchins, and sea star, riddle star. Okay, sea cucumber. So the sea cucumber. Okay, so that's got to be our closest relatives. Okay, then everybody did that. We're talking about, but all the other animals, we're in the chordates, right? Which is all the animals you think of at the zoo and stuff, the birds and all that. They have a backbone and they develop the same way too. But we are closest. You throw out of the vertebrates, which is all the big animals you think of. The weird stuff, the closest we're getting to is the echinoderms. Everybody else, these primitive ones, are called protostomes, and they develop mouth first. We'll break that down for you. Now we'll see that in some of the pictures. But I think it's pretty fascinating. So this invagination leads to, and the different color is showing different germ layers. Okay, and if you remember, there are 3 germ layers that's been on every ACT ever given, right? Ectoderm develops brain and skin, mesoderm is muscle and bone, and then endoderm is the digestive system, guts, and some of the glands. Okay, but anyway, isn't that kind of cool? So this is the theory, and we do match these guys with DNA. So one of the craziest things for me, when I took zoology that's not lie. I'm 56, almost 40 years ago. Yeah, that's kind of scary. Okay, so 38 years ago, we didn't have any DNA fingerprint. And so all the taxonomy was just kind of like, you look like this guy, this long guy. You know what I mean? It was by anatomical things that we could tell, okay? And these stages of development stuff. And so one of the cool things is, to me, is that we look like this. I mean, that we do that. You look like a little spear at this development. But the DNA fingerprinting is now proving what we thought was true, but it's also proved some things wrong that we thought were true. Like who skinned who? And I think we already hit the biggest one was the fact that these, remember the archaeobacteria, they're closer to us, eukaryotes, than they are to regular bacteria. That's a mind blower, okay? That to me, and I mean, for our Bible people, and I go to church, that to me, that is earth-shaking as if we were to find 5 more commandments. Like some archaeologist finds the 10, and there was a slate that Moses dropped. Okay, like the 10 commandments, okay? That would be something on that order, okay? That's earth-shattering to me, okay? That we're kin to this eukaryote closer than to this archaeobacteria than they are to each other. the other prokaryotes. So anyway, the DNA is suggesting this is all the animals come from this guy. The theory there that names it here is this colonial flagellum hypothesis. I'm not going to bore you with it, but this is how it occurred. And then these become specialized, okay? Now, another thing we're going to notice as we run through these animals is we're going to see a shift in body form, okay? And what I mean by that is there are three of these. You can have three body planes. You can have no symmetry, which is going to be sponges. They ain't got no front back. You can't whoop the butt of a sponge, okay? He ain't got no butt. Then you see radial symmetry, which is a circle body plan, like a jellyfish, right? Or pine tree kind of has a circular body plan. I guess you could say trees kind of have a circular body plan. But then the one we got, look around each of us, we've got what's called bilateral symmetry. We have a left, right, the front, and the back, anterior, posterior, okay? And so that is the higher level animals start to do that. So we'll see, I think one of my questions is list the three types of symmetry, right? Asymmetry means no symmetry, right? And then you have this circle body plan, radial symmetry, and then bilateral symmetry. The genes we discovered, these Hox genes, are responsible for this. And so we know that variations in that Hox genes is what made bilateral symmetry evolve out of radial symmetry, okay? So we can see these genes today. So there it is. I'm sorry, I was getting ahead of myself. Sorry. So there we go. Remember asymmetry? Remember the word? I don't know, guys. I don't do English, and I really want to have a meeting with them because I think some of the stuff they do is crack. So A in front of the word means what? Not what's following. So Robin is A tall, A cute, and A thin, right? Okay. And A talks slowly. Okay. All right. So A means no, radial, bilateral. That's cool enough. Another thing we see as we work our way up the animal kingdom is cephalization, means to have a head. So the evolution of the head is a big deal too, with the brain in it. Okay, let's take a look and talk. There's talking about the three germ layers that I've already mentioned here. Here's another cool term. There's my term I just mentioned. Protostomes, that first opening becomes the mouth. Deuterostomes, the second opening becomes the mouth. The first one is the anus first. So we are in that deuterostome, which is anus first, in that invagination. Here's radial symmetry. There's a hydra, which we're going to see in a few minutes. And here is a turtle, which has bilateral symmetry. Okay. And other differences here, I'm going to the picture here, but we'll talk about these. I think we got a good picture here. Come here. Here are other differences between protostomes and deuterostomes. Notice that the protostomes do what they call cleavage. Cleavage is a stage of rapid cell development, like when you go from 2 to 4 to 8, 16 to 16, I get that right, 16, 32, 64, 128. So the rapid early stages of cell division after the zygote forms with sperm and egg is called cleavage. And they have this spiral, or what's called determinate cleavage. You can see it from top and side. And the deuterostomes have cleavage that's radial and intermediate cleavage, how they divide. And here's the big one, anus first versus mouth first, right? And then others show how the coelom forms is a little bit different. Now, the coelom is something that's always been crazy for a long time, but I want to explain it to y'all. The coelom is an internal open body cavity. So has anybody ever like killed a deer and gave the deer? I mean, you know how all the abdominal organs just land in there, and then the heart's in another hole called the thoracic cavity. So that coelom, your coelom is an internal body cavity for all the gut stuff. And a true coelom is an open body cavity that is surrounded by muscles like our owl muscles, with each other like organ maybe have muscle around it like the digestive system. So to have a true coelom, you got a digestive system with muscle surrounding it, laying in a body cavity that also has muscles around it. Our owl would be that, okay? What we're going to see is some animals don't have any coelom at all. Okay. Others have a pseudocoelom, which they have the internal body cavity, but it's not lined with muscle. And then you have the true coelomates, which are us. Okay. That's a lot thrown at you, David. I'm just laying it out there. But those are the main. But the main one, I'll find on the test if you describe them as being that fate of that first pinch in or invagination. Okay. A couple of other things here that's kind of cool. It said first opening. There we go. Okay, so certain protozoans and all deuterostomes have the true. So that all deuterostomes have the true coelom. Certain protostomes have the true coelom or the pseudocoelom. We'll talk about that. I thought I saw another term I wanted to know, but I think we got those there. That's cool. Okay, moving on. Okay, now we're ready. Now we're going to kind of dive into it. So look for these changes in those different things. Okay. Another thing I would say to look for too, just a couple of trends as we do this. How do we get oxygen? Is a big deal. Because some of the simple organisms just do gas exchange. But then we see evolution of gills and lungs, right? And then the other one is kind of cool, is watch that digestive tract develop and look for kidneys. The early organisms don't have kidneys, but then the worms have little nephrodems, which are little kidney-like structures in their segments. And then we get to the higher animals, we start to see kidneys. So kind of look at that. Look at the development of the heart. There's a lot of places you can look as we go through this. Look at the heart development. It gets more complex as we work our way up to the top of the, and I'm sorry, but to the higher chordates, which we are part of. All right, here's our first group. And one of the things that I didn't do here, I'm just going to go over my questions, but I used to love, and I don't think I'm going to do it, but I thought about throwing a phylum matching together here. So I know y'all doing phylums in lab, right? Looking at these and the phylum they're in. And the mater food, which we actually use the mater lab, a lot of it come from that. But they can sometimes you can get differences in taxonomy on this, even amongst the same book company. So anyway, this is phylum Porifera sponges, okay? And they are the simplest animals in the animal kingdom, okay? It is the only level of animals to lack true tissue, so they don't have muscle and nerve and epithelium. They don't have the three germ layers, ectoderm, endoderm, mesoderm. They have a few cell types. They have a sac-like body plan that has holes in it, but they do have a flagellum cell called a collar cell that has flagellum. Take a look at this. Does that not look like those little flagellums we're just looking at? So, and I mean, come on, man, that's the same freaking cell, right? That collar cell has a flagellum, and they take ATP and they whip this flagellum, and that causes water to come in through these pores, and the water then is blowed out the hole at the top called the ocellum. So that would be a good test question. So when I'm looking for multiple choice, if I know we want to say, what's that hole at the top that the water blows out over the sponge, right? Now there you go, excuse me, in Porifera, then that's a cool term, right? Ocellum. I'll be looking for something like that. Our collar cell. Then they have amoeboid cells in here that pick out food particles as it goes through. So they are filter feeders. They filter food particles, sugars and proteins out of the water, and then that amoeboid cell then spreads it, and they spread it out to all the cells by osmosis or active transport. I have had some freshwater of these, and I used to be a real big aquarium guy. Then when I had kids, I lost a lot of my hobbies. Okay, but anyway, one time I had 110 and an 80-gallon aquarium and 10-gallon aquariums. I was into fish breeding, having fun with my fish. But one time I grew, so I had some freshwater sponges, and you would not have to filter the buy as many filters for that tank, because they would filter the water. That's what they are. They're filters. Okay, living filters of this. And this is a very small one here. This is one maybe just a couple inches tall right here, but they can be rather large. They also were broken down, and you can go take classes in this, but they actually are broken down by the internal structure called the spicule. And the spicule is their skeleton type material. Okay, and there are three groups, and I think you're familiar with one of them. There are some that have a silica glass skeleton, which you would not want to grab and rub down your back to clean with. Others have a calcium bicarbonate, like a chalk skeleton, like a calcium kind of chalky skeleton. But the one you're familiar with have this protein called spongin that we use. So we take them and pull them out of the water and dry them out and let all of the cells rot out. And what you see, if you ever seen a real commercial sponge, this come from a living sponge, it actually is all these cells are rotted out except the spicule. And so his skeleton is what we use to clean our clothes, to clean our body with, or clean the dishes with, if you're using a real sponge. Most of these are just synthetically made, quote, sponges that you buy now. But you can go down to Florida and buy these, and they're very pretty, and we got them in the lab, you'll see them, and you can clean with them, okay? But they will also hold bacteria and stuff, so it's not a good idea to keep that one dirty rag in your sink for 6 months. That can get real bad for everybody in the house, right? Because you're just reseeding it. So they actually are broken down by the type of spicule they have. Okay? See if I missed anything else on those. They mainly do asexual reproduction when they fragment off a piece of bud, falls off and becomes a new sponge. They can have sexual reproduction with a flagellum larva stage that swims to new locations, but most of their life they sit in one place, and they're said to be what's called sessile filter feeders. That word means they sit in one place on a rock or whatever on soil, and they don't move. But they have a lava stage that can't free move. Once again, most animals have movement, right? And that's the only place they have movement is in their lava stage. Okay? So you can take a whole class probably at senior college on Porifera. I don't know, not at all colleges. You'd have to have somebody, because this is pretty rare, but there are people, this is their life. This is what they study. Here's one I'm not real familiar with, these cone jellies, and I'll show you one of these in just a second. Look at them right here, but they put them in here with my Nidaria. The cone jellies on the left, and this is a Nidaria on the right. Once again, I want to slap somebody hard, okay? I'm a member of the Drop the Freaking C Society. Why don't we put a C in front of, why is the C in front of N silent? I just want her, I'm not a violent person, but I would like to have that conversation with the English people, right? So I say we either call it scenario or drop the frequency. What do y'all think? Okay, I just hate stupid. I don't do stupid very well. Okay, so it is pronounced, not area. Silent C. You call it scenario, I'm just going to go thumbs up. Okay, I'm good with that. Okay, until they get their junk stripped together. But that one is more interesting to me, and they get their name from them. All of them have this structure called nematocyst. Okay, and the nematocyst is a stinger mechanism found on the tentacles. Okay, let's go back and we'll talk about those. Let me see if we get anything. I don't have much on the comb jellies. Some of them are bioluminescent. They make the light. Don't have much with them. The DNA suggests they are more ancient than sponges. The sponges, they may have been the first ones actually in the animal kingdom, and then the sponges may come from them. They're the largest animal propelled by being of cilia. Okay, so most of us have flagellum or something bigger, but they're the biggest organisms to have cilia. That's their claim to fame. Okay, now they get their name from having this nematocyst, which is a trigger stinging mechanism. So this is a trigger on their tentacles that if it touches another surface, it opens up a hole and lets water rush in, and then this sharp tentacle sticks into the prey. And they use this to kill small like water fleas, or whatever, and they'll eat them and put them in their mouth. They actually have a mouth. So they're going to have a sac body plan. They have a sac body plan, and they have two forms they can live in. The polyp, kind of like a sponge sitting on the bottom of a lake, that's a non-movement stage, but they usually have a medusa stage where, like the jellyfish, where they can free float in the ocean or go where they want to go. But let's look at that nematocyst. So here we go. That's the jelly. You see these kind of look like the jelly cone, the comb jelly or whatever, but they're a little bit more advanced. Okay, here's some examples of them. Here is a jellyfish. The one on the left is Portuguese man of war that can kill humans. These can be up to 20 or 30 feet long, and that's a dead fish, and there's probably a 2 or 3 pound fish in that picture. That is got, I believe that's right. I believe there's a fish in there and it's killed. This looks like a bad blow up of a picture I've seen before where there's a fish, they hunt, they use that tentacle to kill fish, paralyze them, and eat them. And of course, coral that we see at the bottom of the ocean, all of our beautiful coral reefs come from Nigeria, and they all have the nematocyst on them. Cinemes, coral, I'm trying to see, we've got hydra, is a freshwater version. We see our first nerve net, not a spinal cord, but a nerve net. I've got a picture of a hydra coming up. Here we go. So here's a hydra, and he has that sac body plan. Now, by the way, if you ever date a Nigeria, it's going to be really weird if you get real serious, because their mouth is their anus. So that kissing thing can be weird, right? I'm just thinking I'll have problems with that. Okay. So they don't have an anus, they have an opening in the mouth. They bring the food in, they digest it in this gastrovascular cavity. All the cells then have access to this cavity pretty much, and they absorb the nutrients they need, then they blow the waste out the mouth/anus. Should be mouth/anus, okay? Just one opening. But on these tentacles, they have that nematocyst, and so when that trigger touches, water rushes in, and this little thing, like a cloth, like a cloth needle, it sticks out, and that needle has a neurotoxin on it. Now, has anybody here experienced jellyfish? Anybody? Okay, I don't have time to tell my whole jellyfish story, but I'm just going to say that one of the most embarrassing moments of my life was a trip to Florida when I was like 16, and I was out there snorkeling with my dad. We were out on a little boat, and I was diving, and we actually kept running sand dollars, and we're catching these echinoderms. We had a boat full of sand dollars, driving them out, bottom, giving them all our redneck family friends. Okay. So we brought like a 5-gallon bag of strawberry bag of sand dollars. It was so fun. Me and my dad had a good time. But I was like me to use my dad, and he had just came up with a snorkel, and he looked at me and said, "Don't dive," don't dive. And I'm like, dude, we're like 15 foot of water. I knew my dad go, I think I was like shy of a bird, and I dove anyway. So I dove down, and I got a double handful of sea. This is what we were doing. I worked with him all the time on the farm. We would laugh, we would kidding with him. And I grabbed a handful, and then I used the bottom of the ocean to propel off the bottom of the ocean. And when I came up in like 15, 20 foot water, I looked up and I was kissing the mouth or anus of about a 20-foot jellyfish. And he had tried to warn me. And so when I came up, I had about 10, 15 foot jellyfish completely wrapped around me. Everywhere that tentacle touched me, it looked like somebody put me with a big stick. Or what, how many of y'all remember mother whipping y'all and you get the stripes where she hit you? Okay, how many of y'all remember whip? There's your problem. You get in therapy right now. Okay, there's what's wrong with y'all. Y'all one whip. Okay, whipping is good for kids, right? Okay, especially if you're bad like I was. I'd be in park for death from my mom and dad. I'm pretty sure I was a bad kid. Okay, but anyway, So basically, we come out, and at that time, I was very allergic to bee stings and stuff, so I'm having anaphylactic shock. I'm swelling up. And so my dad grabs me, and we're floating, and we're making a scene, okay? And this is right when Baywatch was coming out. I mean, original Baywatch. So we heard them going on, we got drowned, or we got drowning out here in Sector 3. And they're blowing the whistles and stuff and all that. And finally, when I get to where I can stand, I walk, and I'm coming out of the water, and I'm a short, fat dude, and I grab that jellyfish, and I just throw it in the sand, and I pass out. And I hear a four-wheeler pull up, I'm thinking it's Pamela Anderson at 28, not at 68. I hear somebody jump off, and it ain't Pamela Anderson, it's a dude. So I open my eyes, I don't want no mouth to mouth some dude, you know. And I'm like, and he goes, damn, that's the biggest jellyfish I've ever seen. I said, yeah, tell me about it. And he goes, man, he got you bad. And so he reached in his trunk and pulled out a big box of meat tenderizers, like we sprinkle on the steaks. I'm like, what the crap? And he sprinkled that all over my body. And meat tenderizer and tobacco will neutralize the neurotoxin in the metacyst. There's just some stuff to know today, right? And you ever got stung by a bee and had to put tobacco on it? Same thing there. So anyway, it knocked it out, but it didn't take the red stripes. So I had to spend the rest of the week in Panama City looking like somebody beat me. Like I'm telling y'all, everywhere on my body was red stripes except right here where I had that stroke. This was perfectly still ugly with no red stripes. Really bad. Okay, so bad experience, okay? But true story, and luckily my dad was there to help me get out of the water, right? But that's true. The hydra, and they'll eat water daphnia, water flies, and even the big ones will eat a human, will kill a human, try to eat it. That works Portuguese bound water. Next one here, Spirliums, not a big fan of them, don't know a lot about them, okay? Two groups here, and once again, it's a big group here, but the group that I'm familiar with is the protozoans, the flatworms, rotifers, and mollusks. The virozoans, I'm not a big fan of them. I don't know much about them. Here they are. Can't say much about those. They're little aquatic organisms in the animal kingdom. Other than that, I got nothing there. Moving on. But we'll go to mollusks. This is a cool group. 100,000 species, 3 body parts. One of your short answer questions. 3 parts of all mollusks are a visceral mass, that's their internal organs. The mantle, which is a part outside the visceral mass that excretes the shell. They all have a bicep, they have 2 shells, and then they have a foot. And the foot comes out of the shell, and this is how they dig into the soil and the ground for movement. Once again, I think I told you I worked on a project one year where we were living on the Tennessee River, and I got to know these professional divers, and they brought me mollusks, and we would put them in cages to make sure the herbicides and TBA was spraying in the 10-ton would not kill the mollusk and the fish, but kill the plants. So we would sample all 3, fish and plants. And I brought these home. I kept an aquarium with these mussels for probably 5-6 years. And I'm telling y'all, it's the craziest thing ever. You can cut the light off, and you can hear the shells, the gravel rustling, and they would come out, open up, and do their gills and fish feed. And then you turn the light on, and then it all go back under the gravel. Okay, so it's pretty cool. Once again, some cool stuff here. How many of y'all like oysters and stuff? I do. I like me some oysters. Don't eat them raw. They can get hepatitis and all kind of bad stuff. Nothing like going home and telling your parents how you got gonorrhea on the shrimp. That's not going to work. That's not going to fly real good at my house. Like right, and what else did you do? Okay, but anyway, but no. Okay, real quick. This is quick one. I hate to do this, but how many of y'all have ever been to the beach? Okay, good. How many of y'all seen the pipes coming out of the when you walk on the beach and you'll see a pipe going out the ocean? That's the septic line. They flush the toilet up in 18th floor and it blows out to the ocean. The oysters are getting that, and that's how some of these hepatitis and diseases get in the oysters. Get them steamed or fried. Frying kills all that stuff. Okay, and even I like them stacking them steamed now. Or grill them, so they grill them that'll kill the bacteria at least and mess up the viruses. Hopefully. But pretty cool. So we eat the whole thing. So don't ever eat half an oyster. Eat the whole thing. Don't eat half and look. That's probably do you in because you're eating the whole body. Okay. So here we go. Check them out here. Here's their mouth coming in. Food comes in. Look at back here. You can see the shell, gonads, heart. They got all the internal organs. And we eat the whole thing, that whole visceral mass. Cool curd. Mollusks in that group. Kind of cool. They have this open cardiovascular system. So they don't have the capillary beds like we do. We'll talk about this later on, but we have a closed circulatory system. Our blood is always in arteries, veins, and capillaries. The insects and some of these other organisms have an open circulatory system. So it'd be kind of like we had a water hose and spray blood all over internal organs and a big vein down at the bottom there to re-kick it and carry it back to the heart. So they also have a kind of a a different one there than we do. Bivalves include the clams, oysters, mussels, and scallops, right? No head, no regular, open circulatory system. Once again, means they don't have the arteries, they don't have the capillaries. When you get a bruise, you broke your capillaries and you have blood now it's cooling in your tissues outside the blood vessels. That's what bruises are. It just kind of makes that interesting. Cilia, right? They collect the food off that as they filter feed with those cilia. With freshwater clams, those are very tasty too. Here we go. There's scallops at the top. Scallops that y'all like to eat is a bivalve. They pull them out of their shell. Mussels. We did go to Florida last year and rented this big house. My entire family, my mom, all my brothers and sisters. My dad's not doing well. He started getting dementia. We tried to spend more time with him. So we went down there and rented this like 2-story house. And I went and bought this steamer thing down at that place down at the store. It's called steamer, and you buy 100. That's $80, and it's like 20 to 30 pounds of all these, like the whole family. Every bivalve you can find down there, right? And the lobsters and all cook the whole thing together. So we had like a little biology class down there, opening us up and eating them. But it would scare a little kid. Little kids see them start, oh my gosh, don't eat that thing. It's looking at you. Well, it is what it is. Same thing here. You can see the same body structure here. I do find this a little bit sad. The in current is coming out here, out current here for water, and then they poop right on their X current, right above their X current here, which I wonder if there's a little reflow there. I always wondered about that. See if I circulate a little of that water back. If you ever tried to open them, and that's what I did, I actually grilled some, and I bought some that had not been salt. You used to have to take a knife and cut these two muscles, anterior adductor and posterior adductor, and then pull it open, and I grilled them over the grill. Pretty good. First time I ever grilled them. Some oysters. All right, gastropods are slugs, snails, conchas. Anybody eats the snails or slugs? You ever seen that? Some of those guys are escargot, right? Well, if you ever take a cruise, they like to do them one night. So they'll have a hors d'oeuvre with escargot, and they cut them and cook them in these little ceramic things that just have a little hole in the shell set in. So they just smoke those, cook those little rivers in the oven. And you get a little fork reaching there and pull them out. Really kind of fun. It's kind of, once again, I like exploratory food where you got to tear something up to get to it. Because you have to little reach in there and pull them out of the shell. I wouldn't go order them, but they were part of the deal. I think they're that good. Okay. See if we got anything on those guys. Of course, these are the ones that y'all evil people pour salt on and osmose them to death, right? You know, you've done that, right? On little slugs and stuff. But here's one in the shell that they like to cook and pull them out. Cephalopods, the next group here of squids and octopus. Very interesting group. They have a beak in there. So if you open it to the very inside of that, when all the tentacles come together, they have a beak. And the octopus has been shown capacity to learn. The two things that stuck out to me about these is they have these tentacles with suction cups on, and they also have an ink gland. Have you heard of this? So when they get scared, they can squirt this. It's not really ink, but it's black ink. It's like ink. It's squirted in the water. It makes the water. They can one octopus maybe as big as my leg to squirt enough ink to make this room jet black in the ocean. So they squirt that when a predator is attacking them, and the whole thing turns black. They just swim away. They actually use jet propulsion. Now they don't have jet fuel and ignite, but they blow water through a small hole, and that's what jet propulsion is. So jet engines work by igniting jet fluid, and the exhaust has to blow out a little hole, and that gives them the thirst to move the plane. So these guys use water and jet propulsion to swim. Very cool. And I actually had an experience. I had a moment with one of these. I'll tell this real quick. Kind of a cool story. Here's a nihilist in this group too, and here's a squid. And we use these a lot for bait here. Go deep sea fishing. But me and my dad were deep fishing one time, and we did a 12-hour fishing trip where you drove out 4 hours, fished 4 hours, and 4 hours back. And they had a cooler about 2 times the size of that podium, and they had a sign that said the fishing trip's over when the cooler's full. And me and my dad laughed. And we've been 10 or 15 times and never filled a cooler. Matter of fact, I took him last summer thinking it might be the last chance to take him, and our boat caught on fire. No, it's crazy. And I have my daughters there, and I think, I don't mind dying with my dad today, because I'm not having a good life. But I got both my daughters and my son-in-law, and this boat's on fire. And the captain was an idiot. I think my dad with Alzheimer's could have done better fixing that engine. I mean, really, he got on fire. And it was so bad that when we got back to dock, I demanded him give me some money back. Because I paid him in cash, and he gave me like 70% of it back. So we had a pretty good boat ride for $500 or something. But he did give me some money back. But one time on a much better trip, I caught a octopus. So I snagged an octopus. It was probably about a 3-foot octopus. And we put it in a live well on that fishing trip that we did for the cooler up. And so after we were moving and went fishing, I had to, I was already knew I was going to be a teacher at this point. So I was probably, still in grad school, but I knew this is what I was going to do. I got to do this to tell all my students if this is really true. You read stuff in books, you go, is it really true? Is there really suction cups? Do they really have that ink gland? So I reached in there and that dude wrapped all over my arm. It took me 20 minutes to get my arm. Like suction cup. I mean, so check suction cups, they're really there. That little tinkies on my arm. So then I said, I got to see the ink gland. So why am I going to get in front of the ink gland? I'm going to have to scare you. So I went, boom, that didn't work. Got to tell you the scary story, it didn't work. So I took the oar and slapped the heck out of him. That worked. So I had to attack him. He squirted that ink, and the captain's like, what are you doing, man? They had to run water through there for 20 minutes so they could get the bait out of that live well. I mean, it turned it black as dot. I mean, it's my pants. Crazy. So check off on that. And we got through. He cleaned it and asked if he could have half of it. And I said, you can have whatever you want. I'm in your boat, sir. But it was like lobster meat. The whole tentacles were lobster meat. Very expensive if you try to buy it. Octopus is pretty good eating. We ate some of that. Took some of that on and ate it. It was very, very good. Very tasty. Yes, we killed him. Sorry. I come from a very strong hunting background. I probably have about 30 guns. I've got a gun almost every Christmas since I was 6. I like guns. I'm fine with guns. My wife scared of guns. It's so bad. We don't even talk about it. The guns let over and she just pretends they're not there. No, but they're there. Okay, anyway, so let's go to worm. Amulets. Very cool one here. So we got several groups in here. The big one here is a liquid keys or freshwater, and have a few of these seed up. And the polychaetes are marine or ocean saltwater worms. And if you ever seen them, they have these great big parapodiums that look like oars that they crawl with. The CETA is the little bristles on an earthworm, and that's how they move through the earth. So they have little fingers, little extensions that they can move, and that's how they move. Okay. We have a good spinal cord, nerve cord, closed circulatory system like this. Now we got capillary beds, well-developed digestive system. And I think I had a question about the 3 types of skeletons, right? Okay, y'all know endoskeleton is us, right? Bones, internal. Exoskeleton or what? Insects. Come on, how many of you ever had like a bee? I'm not the best. I mean, anybody else like me that don't clean the car very much? I'm not a big car clean person. And so I'm just saying, I had a bee one time that got stuck up in the windshield, where I couldn't reach it. And I just left it there, and he looked perfect for 10 years. Because he died right out internal with the exoskeleton. This is why you did an insect collection in high school. You can go out and collect insects, they'll look the same the day they die. I always thought we should do like a vertebrate collection. You go get a squirrel, and a possum, and neighbor's dog, and neighbor's cat, and put them on like boards. That would be bad. It would be nasty smelling, they'd rot out, and you'd have a skeleton eventually, right? Maybe a skull thing, and then that turns out that we don't do that. Okay, we kill the insects. There's more variety in insects. Okay, but the sin is kind of cool. But we do know that about liquid keats or freshwater, polychaetes, earthworms. We'll get to them. I'll show you one here. Rings, segmented rings. They're hermaphroditic. They have both male and female reproductive system in their bodies, so they're hermaphrodites. So when you see them laying next to each other, they're having sex. That's some earthworm porno for y'all. Okay. The patellum is their reproductive structure. That's where they swap the sperm and eggs, or the sperm. The sperm is what transfer. Once again, they lay down next to each other, and each person gives the other one, each worm gives the other one sperm. And that's where it goes through in that structure. They have a head with a mouth, so they have cephalization. We just don't really notice it. And they have an anus at the end, so they have a complete tube digestive system. They have a 5-paired hearts, 5 little structures, 5 ventricles that pump the blood. The cropping gizzard would be a storage area that crushes the food, right? Kind of like our stomach. And the gizzard, they like to have a little grid in there, rocks to help grind the food up. Okay. And then each segment has this is the kidney structure here called the nephridium. This is each segment has a little kidney structure. Here's the digestive tract going down, that's coelom, and here's the spinal cord, sit there. The CETA, I don't see a good picture of it here. These are the little, there they are, little tentacles that stick out, and that's how they move. They move them, and that's how they squeeze through. Because they have that hydrostatic skeleton, they have water skeleton or blood skeleton, they can squeeze through pinholes, the whole body. So you can't get out of window this, and we can't go through a hole that's shorter than your shoulders, right? They got to go through it, right? So it's hard to get through small holes for us. Okay, and then of course exoskeletons are the insects. But that's a cool, quick answer to that one. Is anybody? There was a company a couple years ago selling these. Now, how many of y'all seen this? I was ahead of my time. How many of y'all seen the veggie burgers they got now? You know, the Whole Foods, or what is it? There's one of them, not Whole Foods. There's a company of, oh my gosh, I've been watching the stock. Stock went from 20 to like 300. And they're making synthetic sausages made out of plant. And they figured out how to make a sausage link or a sausage that looks and has the texture of pork, but it has no grease in it. It's all plant-based. They even found the color of like hamburger meat, even the blood color, and they can make hamburger patties out of pretty much out of protein, plant protein. Well, before this, there were people growing earthworms, who, by the way, which are very good, clean, white meat. They're very good eaters. They're not poop eaters. You know, they're not, anyway. Not like some other animals we like to eat the crud off the bottom of the lake, which is not real healthy, right? Okay, like the oysters and stuff. They're filter feeders, man. They're poop eaters. Okay, these guys eat clean meat, and they were grinding them up like a hamburger. You can order quarter pounders, and we can have a grill. I always want to do that with my class. This is how I'm going to my house. I'm going to cook worm burgers where I have we're not eating a worm burger. Now, see, y'all are being ethnocentric. Y'all are going, I'm not eating a worm, but you know, if you ever see them process a cow, I go with the worm. Okay, okay. I go with all of them. Okay, I'm non-discriminatory on that. I'll eat anything. Okay, but I would like to try the earthworm burger. I tried to get my wife to give me the sausages. They don't have them here sent over in Hernando yet.
The cauliflower burgers are disgusting.
Okay, yeah, y'all had the Whopper. The Whoppers they got a Whopper now that's made out of the fake meat. But for the true animal lovers, right, we should eat. You know, y'all don't believe in killing any animal, right? Okay, so it's okay to kill a tree or eat a plant, right? So, but if we can make some sausages that taste, I don't even need the flavor. I'm a texture eater. I like the texture of sausage. So if we can make that in a plant and give me some fake flavors, I'll try it. Leeches are used in hospitals, right, to suck fluids out of wounds after surgery. So they're very sterile leeches, but they actually suck blood, and this is used after surgery a lot of times. I've also read where when they like reattach fingers and stuff, they'll put leeches on them because they help keep the swelling down. So leeches fall in this group. They have the sucker. Filamentes is the one that has the acetylate. They don't have a coelom at all. You can't gut these guys. I mean, you can cut them, you'll never find a body cavity. But look what he does have. He has a endocrine system and an excretory kidney structure. Here is the reproductive structures with both testicle and ovaries, so they're hermaphrodites. They have a soft body plan. They bring it in, this pharynx is digested, blow it back out. They also have a nerve net. Okay. And I think the coolest thing is that we have a nerve ladder, we have a nervous system. Look at that. Nerve ladder there, sort. They have an eye, have a head. But I got to tell you my favorite thing on these guys. We're about done here. We're almost got the chapter done. But I actually worked with a guy that was a specialist in planaria. So when we got the microscopes, he brought planaria out for them to look at under the microscope. That's what his PhD was in. He was a planaria expert. So we get through the day, guys, named Dr. Columbus, and I think he's passed away now. And I was working on this when I first started teaching lab. And he told me, Mr. Robison, go get that big planaria out of that bowl and bring it over here. You get a knife. And I do it, and he said, now cut it in two. I'm a hunter. I don't mind dove hunting. I deer hunt. I've killed a lot of animals, but I'm not going to kill this planaria just for no reason. You know, why? He goes, because I want you, because I told you to. Okay. It's your job. Okay. So we cut it in two. And he said, now put the tail part in another petri dish, leave the head in that petri dish, put them back in the incubator, and let's look at them in 2 days. I'm like, well, you got dead planaria. The head grew a tail, the tail grew a head. These guys have unfreaking believable genetics of reanimations, whatever, of regrowing body parts. You can't do that with many animals, right? You cut your dog's head off, and the dog is dead. Okay, is that clear? Do not go home and try. I just got this vision of one of y'all going home and trying it on the cat. Don't do that, okay? You killed your cat. You do when you take your goldfish and do that, and it's dead. Okay, fish is dead. These guys can regenerate body parts. Look, I don't know if we got any sports fans in here. Remember Larry Zonka, who was a great running back for the Miami Dolphins, whose son was a great college player. He broke his spinal cord like his senior year. They're researching these guys at his institute in Florida. He died last year. He raised hundreds of millions of dollars for spinal research. He was gonna have his son walk again. His son's still alive. His son's about my age. He's been paralyzed since college. But they've done a lot of good research, and they're studying these guys because they can regrow their spinal cord. They can regrow their head. So there's some genetics here we need to explore, and maybe one day we hope we can reconnect spinal cords. We can reconnect nerve fibers. We can reconnect fingers and toes and stuff, but we can't do the head and the eye yet. Okay, but research is going on, and these guys, we can learn from these guys. And I actually ended up cutting it into 4 pieces. And I think I did it down to 8, and then we put it in the blender and killed it. I mean, there's a limit to what it can do. You chop it up, she's giving me the evil eye. Like a loaf of bread is dead. Okay. All right, then they're hermaphrodites. I think I'm going to stop here. It's time. I think we almost got through this. We like a little bit here. I'll tape a little bit for it. But we got some flukes and stuff that are parasites. Y'all have a good day. We'll finish it. I'll finish it in my office, or we'll finish it tomorrow in class. We got plenty of time. Oh, sorry about that. We got down to about slide 44. Yes, ma'am. Just let me stop this. Stop the class.